Welcome everybody again to another episode of the ideaXme show that profiles the humans behind the really big ideas that are shaping our world, inspiring the future, and for all those that like really great stories. I'm Ira Pastor, your life sciences ambassador, along for the journey today. We have a fascinating episode for you today. As a little background, the sixth mass extinction is a term that is used for an ongoing extinction event, a species that is currently occurring in the present Holocene epoch, sometimes referred to as the Anthropocene, as a result of human activities. The included extinctions on this list span numerous families of plants and animals, including mammals, birds, amphibians, reptiles, and arthropods, and ultimately with the widespread degradation of highly diverse biodiverse habitats, coral reefs, rainforests, as well as other areas. The current rate of extinction of species is currently estimated somewhere between 100 and 1,000 times the natural background rates. Really amazing numbers. This Holocene extinction includes the disappearance of large land animals known as mega-fauna, which comprise large or giant animals of an area or habitat. Among living animals, the term megafauna is commonly referred to these are the largest extant terrestrial mammals, including elephants, giraffes, hippopotami, large bovines, and rhinoceroses. I'm honored today to be joined by Professor Thomas Hildebrandt, who heads the Department of Reproduction Management at Leibniz-IZW and is Professor of Wildlife Reproduction Medicine at the Freie Universität Berlin. Dr. Hildebrandt studied veterinary medicine at Humboldt University in Berlin, received his doctorate summa cum laude from the Freie University of Berlin, and is certified as a zoo, wildlife, and game veterinarian. In his extremely specialized field of reproduction biology, he's one of the pioneers of assisted reproduction in large mammals, including elephants, rhinos, big cats, and panda bears. Dr. Hildebrandt has represented a number of professional societies, honorary professor, fellow of life sciences at Melbourne University, research associate at the Smithsonian Institute, fellow of the Zoological Society of the San Diego Zoo, scientific associate of the Taronga Conservation Society in Australia, conservation fellow of the Zoological Society of London, and veterinary advisor for the European Elephant Taxon Advisory Group. Professor Hildebrandt, thank you for taking the time out of your schedule to come talk to us for a while.
Yeah, hello.
Great to have you. Could you start off, we typically give our guests the floor for a little bit just to talk about themselves. Could you give us a little bit about you and your background, where you grew up, how you got interested in this unique area of zoology, veterinary medicine, and how you sort of have arrived at this highly specialized area of reproductive biology in large mammals?
Yeah, I can try to give you a little bit of an insight to my development. I grew up in East Germany, in Berlin, and went to school. And with an age of 6, so right in front, beginning of the school, the dream came up to be a veterinarian. That never lost me. I was always under the impression that is the right profession for me. Mm-hmm. So I did everything to achieve this goal, and it was not so easy under the communist time in East Germany. I was quite a free-thinking person, and the consequence, I was banned from university for 5 years. I worked in a dairy farm to gain some points for the system so that I maybe had the chance to enter university again. And then finally, I got the that took, as I say, 5 years. And then from there, I was so into this dream to be a veterinarian that I was then quickly the best student at the university, achieved a lot of awards as student. And the same people which banned me before tried to utilize me. So that's a story about my veterinary career, at least. And then the Academy of Sciences came to the university and looked for new promising scientists, and did a kind of headhunting at that time. So I came to this institute. It had a different name at that time. It was a Forschungsstelle für Wirbeltierforschung, means a research station for vertebrates. And the director, a very famous zoo director, Professor Date, he was really a person who supported me personally. And so I switched my view on classical veterinary activities to wildlife, which I never had in mind when I came up with a dream to be a veterinarian. I want to be a countryside veterinarian. So I was then exposed to wildlife and then extremely impressed by the variety it provides us, and also impressed how little is known about, for example, reproduction. And it started with the female elephant reproduction organs, which were at that time, it was in the late 80s, beginning 90s, it was totally unclear what happened during mating in the female elephant, and for example, that the hymen stays intact during mating and ruptured only with the first birth. And based on that, my background is actually an engineer family. Everyone in my family is an engineer. I came up with technical solutions to develop an artificial insemination system for elephants, taking account these very specific female reproductive organs. And from there, it evolved more and more. I used a lot of imaging techniques. I got my own department at the institute. I stayed together with long-term colleagues for now over 30 years. We are focusing on different problems in wildlife reproduction together as a team now for more than 30 years. Worked on rhinos, elephants, giant pandas, but also on giant salamanders, naked moorrats. So it's quite a big variety.
Fascinating. I appreciate that background, and it's extremely interesting. When you talk about some of the species that you've been involved with, I know now that you're doing a lot of work with the northern white rhino. Before we get into some of the different technologies and tools that you're using for these initiatives, could you just talk a little bit about how sort of the area of, I don't know if this is the proper term, sort of de-endangering of species sort of works? I mean, in the sense that obviously it's a very specialized community, but how do you go about sort of picking the species? Because, I went online and I looked at sort of the list of all of these animals that are in, I guess, the highly endangered group. How does one pick a rhino versus a species of orangutan or a whale? I don't know if you can endanger whales, but how is this decided? Is there a network of special researchers like yourselves? Governments get involved? I mean, what's involved with this?
Yeah, in the beginning, I think I have chosen my objects, the species we work with, and mainly by interest, on a personal interest, like elephants. I very closely linked to elephants because they are highly intelligent and they are very impressive animals if you interact with them. However, over the last 10 years, we're really focusing on repairing habitats, ecosystems, because in your introduction, you mentioned that we have an extremely accelerated loss of biodiversity. And you can imagine, if you take out the key player of such a very fragile and complex ecosystem, we call it the vortex effect, is tremendous. So if you want to fix an ecosystem, an important ecosystem, then you try to fix the cascade. And you would not start at the bottom, you would start on top, because that's the most critical endangered species, like northern white rhino, which live in the Congo Basin. And we had in the beginning the impression that it's really sad that we lose these animals because they are creature made in a process of evolution, and they didn't fail in evolution. They are only not bulletproof because people poach them.
Right.
But now we have to really put that in a bigger picture, and I think everyone will understand this bigger picture because of Corona crisis.
Sure.
If you disturb a fragile, very complex ecosystem, then you provoke a disturbance which really is a source for newborn diseases, which are very dangerous for the humankind. We're losing our bioservice, clean water, fertile soil, clean air, and we have to fight with germs which are normally hidden in the jungle and which comes out because the bats fly out because they don't find the insects anymore which lived before from the feces of the rhinos. We have suddenly bats with very nasty viruses or bacteria because they look for a new environment because we destroyed their home. And I think to put that in context makes our work more important and also puts the responsibility of what we do on much wider shoulders than only some crazy scientists, because we see what kind of economic consequences this system has, or this dysfunction of the system has. And therefore, we are looking, coming back to your question, for the key elements of an ecosystem to keep it stable and to keep it as complete as possible. And if we take care of the major player, the chances that we achieve that is much higher than if we focus on the side species.
Gotcha.
We also work with naked murrets, which are not the key species in, for example, in the arid environment of Ethiopia or northern Kenya. But these animals represent some unique adaptation, are very helpful for us as humans. They could serve as a new invention or provides new invention for the medical system. They don't get cancer. They live over 30 years. They really have very minimal levels of diseases. So these, the first interaction, very ugly looking animals, is actually a master of disease prevention. And I think it is also important to get that to the young generation, which are more open for these ideas and asking question, where's the sustainable use of our planet, to show them these creatures of evolution, that they can provide solutions for our own future. And it is very wise not to destroy our planet in such systematic way like it happened over the last decades, to take care of these creatures and try to study and understand them.
Outstanding. I couldn't agree with you more. That's very, very elegant the way you put that, and you know, the connection to with the COVID example is quite prescient and eye-opening in that context. One of the, you know, you have this fascinating review that you recently wrote in the Journal of Zoo Biology. It was entitled "Rewinding the Process of Mammalian Extinction." And in this review, you talk about the principles of conservation by cellular technologies. And you create this roadmap talking about how we can use some of these tools to, you know, look at an extremely endangered species and, you know, help them, you know, help create self-sustaining populations. Could you take us on a little journey? Obviously, you know, topics like IVF and cloning, you know, they go back several decades. However, you know, we are now in an era of stem cells, induced pluripotency, all sorts of other interesting technologies that you write about. Can you take us just on a little journey here and sort of where you see the technological toolkit evolving in your work in terms of some of these larger mammals?
Yeah, first I have to say that we developed this technology to maybe rewind the process of extinction or prevent animals from extinction. However, it is a very expensive and very time-consuming process. And it is much better not to fix too many species. So I think it is not a solution to make bad decision or revise bad decision. So in the future, we hope that we get less to do. However, right now, there is a lot of critical endangered species which have this key role in ecosystem, like the Sumatran rhino, the northern white rhino, there are orangutans, you mentioned them. So there is a lot of these key species which need our help. Mm-hmm. I think it is more wise to spend human resources for fixing our planet than to explore the Mars, because that is our environment, our Earth. And if it is destroyed, I think future generations have nothing to live on. And it is, therefore, it is so important to act now when we still have the chances. And this acting is now much more efficient with this new technology. It is really a mystery that the design of every organism based on cells, and every cell has a blueprint inside to make an organism. Mm-hmm. We don't understand why this design is existing, because if you don't use your cecum or you don't use your gallbladder, then it goes away. Organs or a parasite really has only organs, but no other really other organs like feet or something like that. So in nature, we have normally a very strong reduction of things which you don't need. Every organism is made out of cells, and every cell has a blueprint to make this organism. And by the ingenious invention of Yamanaka, sure. We can activate this blueprint, which is hidden as a kind of cryptic information in form of the DNA, and activate that and bring the cells to a stage that it is an embryonic cell again, and then it can evolve from there. You can make by cloning a new organism, you can make by transformation, you can make sperm or oocytes, you can make stem cells for blood production. So it's an incredible way how we can manipulate originally one-way road cells like a skin sample or mm-hmm. on the mouse. These cells can be reprogrammed and start with the embryonic history of themselves again. And this allows us to act in a conservation field in a totally new way. I think we were one of the first who came up with this idea to utilize this technique because it fits ideally to the strategy to involve deceased animals, which are already gone, but we have still some cell material from them. We can include them into the propagation process. So that the argument which often comes up is that the gene pool is too narrow of these remaining few individuals. That's right, but if you include animals which are already dead, which and we have material banked in cryobanks, then our entire population is much bigger than we actually have on living animals. We can have sperm cryopreserved, we can have oocytes cryopreserved, which is a little more tricky, but putting all these material together and working with the high-class stem cell researchers, conservation can much more do than it could do maybe 20 years ago. And you mentioned cloning, because cloning is a word which really has a very negative image. And it is not really a suitable technology for conservation because you produce only copies of something which already exists.
Sure.
It is a technique which is sometimes necessary for doing a part step of the entire program, but cloning is not a technology we rely on because that is not what produces variation, and variation is the basis for surviving. And therefore, we use the techniques to make stem cells, and out of the stem cells, gamete-producing cells like spermatogonia or oogonia, which can be cultured in vitro, and then using their products to make offspring. And this entire concept sounds very futuristic, but our partner, consortium partner in this process is Katsuhiko Hayashi.
Okay.
Could demonstrate that very impressively on a mouse model, and he works now with his colleagues on the Rhino project. So I think if we can utilize these emerging technologies, which are mainly derived by regenerative medicine, human medicine, if we can utilize these technology jump for conservation, then we still have a chance to save species which would be absolutely unsavable in the past. And that is something which drives us. And the way how to achieve it is we're working on a very flexible and very inspiring team of international experts. Everyone came together on a will to do something, even if they had nothing to do with actually with wildlife in their past, but they have a very special knowledge which can be utilized for our goal, and we orchestrate that a little bit, and I think we will have very soon impressive results which can demonstrate that this is the right way in a very elective species, because it makes no sense to apply that on a broad scale. It is very expensive, it is very time consuming, and it is also stupid to burn so many resources for a lot of species. We do that for the mystics which were done in the past. We hope we have not to do that for the future mystics performed in the next decades.
Excellent points. Excellent points. The other, another fascinating component of your work, as I'm reading through it, is, on one hand, you have to utilize and perfect these novel cellular technologies, but then you need to make the jump to dealing with these large animals and impregnating them and controlling everything that goes on with regard to how one impregnates a female rhinoceros that weighs, I don't know how many thousands of pounds, but there's this anecdote in one of the articles about how, unlike how we think of traditional reproduction, in this particular case, you had to not go through the vagina, but through the anus, through the intestinal wall, and into the ovaries. Any other interesting stories about, when you're dealing with a rhino or an elephant or a giraffe or whatever, how reproduction as we normally think about it needs to be varied a bit in the lab?
Yeah, you brought up a very important point, because you can have all these ideas how to save an animal, but there is an element where you have directly to deal with your patient, and the patient is more than 2 tons heavy. It doesn't fill out a survey, what kind of diseases it had before, has it smoked, had it a tumor, nothing. You have a kind of black box. And if you are too enthusiastic as a scientist, you could be easily the one which tried to save a species and kill it with an anesthesia. So I think this burden is very heavy on our shoulder, and therefore we really perform such procedures in a kind of army-like style, so that everyone knows exactly what to do. We are always aware that the procedure could be instantly disrupted because of some life signs are not okay, and because we monitor our patient very closely. So it is a very stressful time when we actually work with our patient. When we do the oocyte collection on the last 2 remaining northern white rhinos, and we operate 2 meter inside with a needle, which easily could slice up a blood vessel, and then the animal would die, then you can get a little bit of the environment, how stressful these procedures are.
Sure.
Lucky-wise, we never lost an animal. We are very precautious about every step we perform. But it can happen that due to some unknown disease, some unknown pathology, that this animal suddenly dies and you are responsible for that. Mm. However, we still have a kind of contingency plan. We would harvest instantly every material which we can get access to. For example, we would harvest the oocytes from the ovaries, we would harvest stem cells from the bone marrow and all that. So that is the backup plan for if things goes totally wrong. Mm. And in addition, we really have to deal with animal welfare. So what can we perform to save a species if this is in context with welfare? Can we, for example, kill a mouse to save a rhino? Mm. So a lot of big question in a room, which has a lot to do with ethical questions. Sure. And we have special experts in our team from the University of Padua who are helping us to solve this question. First, to make aware that the intervention is risky, what to do when something goes wrong, if the animal breaks a leg, can't be fixed. We have to euthanize the last female because it is full of pain. So it shows you a little bit of these side activities and brainstorming to understand it's not always a heroic job. It's quite a lot of details which need to be answered or to be prepared for. And we hope we do it in the best way it is possible, and that we at the end, with this kind of master plan behind us, will be successful. But it is, the direct animal interaction is not as relaxed as the work in the laboratory.
I can imagine. And I take my hat off to anyone that tries to control a 2-ton rhinoceros. But yeah, it's amazing. Really amazing stuff. Coming from multiple ton animals down to something a little smaller, you mentioned the naked mole rats before. I read you have this fascinating review in American Physiological Society journal. It's entitled Neoteny, Prolongation of Youth from Naked Mole Rats to Naked Apes, where you discuss this principle of highly social animals like naked mole rats and humans, which have this prolongation of youth. Evolution hasn't selected certain things away in that context. I talk of aging and healthy longevity are themes that I touch on a lot on this show with guests. I know this is a little separate from your de-extinction work, but could you go into this and a little bit of what you're learning on this particular front? Because it's as you pointed out, this is the naked mole rat, though not attractive, is clearly a evolutionary wonder in its domain. And please teach us a little bit what you've been learning here.
Yeah, I can give you the background why we ended up with 400 naked mole rats and what was in the beginning the research goal.
Sure.
I had an internship at the National Zoo in 1995, developing a technique for artificial insemination in elephants together with our colleagues from the National Zoo. And they also had a breeding station for naked mole rats. And they asked me if I could do an ultrasound examination of the queen. I was not aware of social animal at that time, and that only one female produced all the babies. So I did my first ultrasound in a naked mole rat queen in 1995. And that really was an imprint in my memory, and I always had the desire to have an own colony of naked mole rats. And when we produced an elephant baby for the Rio Grande Zoo in Albuquerque, they had an additional colony which we got for the producing an elephant baby there, and I imported to Berlin. And we start to propagate our colony from one colony to the next colony. We had, in a relative short time, several colonies, and we monitored them with a very specific ultrasound system. And the idea was actually behind that to utilize a mechanism of a naked mole rat, which we proposed would be in place for the rhinoceros. Now it gets a little bit screwed, but I will try to explain that quickly. So if you have a critically endangered species, if you try to use forced reproduction, it would be wise to produce more females than males, because semen can be put in aliquots, and so you need more females to increase the number of newborn than males.
Sure.
And there's only one technique right now, a commercial technique, where you can separate sperm in X and Y bearing sperm. It's a commercial system. It's a flow cytometry, which is monopolized by a company. At that time, it was XY company. And we tried to buy a system from them to utilize it for elephants and rhinoceros, to actually do that, to produce sexed sperm, like you use it now in a livestock industry. For cattle, you can have sex sperm, then you produce male offspring or female offspring with 95% guarantee. Unfortunately, the company was not willing to. We bought a machine, but we didn't get the license to utilize it, which was on a monthly basis $5,000. We had to give back the machine, which we actually purchased a month before. And then I came up with the idea that a naked mole rat male, the pasha, has actually no interest to produce too many male offspring. At that time, it was my understanding of naked mole rats, because I thought if he produced too many male offspring, then he would be in conflict later on when these boys grow up and challenge him. So it would be better to produce more female offspring to enslave them in a colony and make sure that nobody is challenging him. That was the idea why we started with our naked mole rat colonies. However, it turned out that this is not the case. So the sex ratio in naked mole rat colonies is 1:1, and the male and the female offspring, which are in the caste system, not in the reproductive caste, they are absolutely uniform. So there is no sex difference in their transcriptome pattern, which is quite interesting. So they are really neutrons. So that there's a depression, the social depression by the pasha and the queen is so strong that these individuals didn't show any transcriptome differences. Our original idea to utilize a hidden mechanism which allows the naked mole rat to produce more female sperm instead of male sperm was not real. It shows how science is. However, we found a species which is actually capable to do that. It's a pygmy hippo. The pygmy hippo is a small hippo-like species, but has nothing to do with a normal hippopotamus. It's in a different genus, and it lives in Liberia and Nigeria. It's critically endangered. It's a very hidden species, nocturnal. And lives not in the water. It lives in the riverbank and goes sometimes in the water, but it does not live permanently in the water like the ordinary hippo, the big one. But this species produce in captivity, but also in its ejaculate, a clear shift in the offspring ratio, so that more than 60% of the offspring are female offspring. And also the same is in the ejaculate. We don't know how the male manages that, but by providing the female with more female sperm and having an agreement of the female, so there's no counteract, evolutionary counteract inside the female. So the population ratio is heavily shifted to the female side. So there are more females than actually male pygmy hippos, which is quite an interesting mechanism. know something like that in Drosophila, where you can have a lethal mutation in a male or female chromosome, so that all male sperm or all female sperm are gone or infertile. But in the pygmy hippo, it's clearly a shift of fertile sperm. We don't understand that. We published it, but pygmy hippos are very difficult to study. First, they are critically endangered, and also they are quite aggressive. The anesthesia is a challenge in them. We tried to work with wild ones, and that program was disrupted because they are very intelligent. They never entered the traps, so we could not do electroejaculation and study them.
Fascinating. Completely fascinating topic. I mean, it's just mind-blowing the things that you can learn, especially when you're dealing with I'd heard the term pygmy hippo before, but I just wasn't aware of. But some of these, as you said, these niches that teach us these things. It's just really amazing insight. What's next?
We should have a lot of respect for the creatures of nature. Oh, I think that shows us that there are things outside, and evolution is so surprising sometimes that it should make us more devote to our other creatures on the planet, and not thinking that we rule the planet in a way like we did that in the past. I think that has a very short end, and if humankind don't understand to live in harmony or in kind of synergy with these other creatures, then next generation have a lot of struggle to survive.
Sure, absolutely. I'd like to get your thoughts on I know that, this is one shift over, but several months ago I had George Church on the show, and obviously he's very interested in a lot of different things, and one side interest of his is de-extinction, mammoths particularly. Love to get, while I have you, love to get your top line thoughts in general on the theme of, obviously you're working with the extremely endangered, but what are your thoughts on de-extinction? And have at it with, and go in any direction you want with that one. Love to hear your thoughts.
Yeah, that's quite interesting question. De-extinction, the entire development is very interesting for us because it fuel a lot of technologies which are very helpful for what we try to do. And the idea to recreate the mammoth or the Tasmanian tiger, people to the table who have a lot of money and they are fascinated by the idea to have this kind of scientific exercise supported, so that the progress in technology, at least that was the case 5 years ago, was much faster than what we tried to achieve with this program to save critical endangered species. So we lined up with a South Korean team led by Professor Wang. We went together to Borneo to look for pygmy elephants as a potential oocyte donor, because everything is a little smaller than a normal elephant, and also a potential recipient for an embryo.
Okay.
But my heart, if you would say, I could decide how to spend money, I would never go the way of de-extinction, because that is really scientific exercise, has nothing to do with conservation. Okay. It shows what humans are capable of, or potentially capable of, but it has no value for our planet. It is like making a Disney animation animal, and what you do to this poor animal, which does not belong to our planet, a Tasmanian tiger or a mammoth, they don't belong anymore to this planet, and you will never be capable to restore a viable population. So it's purely an exercise, sensational, and is nothing we would really fully work with. So it is an interesting field, and some of the technological approaches are very interesting for what we plan to do or what we do. And George Church is a pioneer in, for example, CRISPR-Cas, the idea to gene editing the Asian elephant genome to make a mammoth. Yeah, it's a lot of money, which could stand in our view in a totally different way, and to help to restore heavily disturbed habitats, even buying land or even use high technology like we proposed with the stem cell technology for the northern white rhino. That is something I personally would like to see, but I totally understand that the scientific challenge is very stimulating, and people like to do that.
Yeah, well, I appreciate your feedback on that. I really respect that. Coming back to you now, Professor, we typically wrap up the show by asking our guests about important influencers throughout their career. Obviously, you've had the opportunity to meet many fascinating people in all aspects of wildlife conservation and zoology and regenerative medicine. So any folks that you want to highlight at this stage, shout out to, that really kept Professor Thomas Hildebrandt on this path, and if it wasn't for them, you'd be doing something else at this point? Anyone you want to highlight?
Yeah, for example, the author Adams, who wrote The Last of Their Kind, I think that is something very pioneering in thinking, and he, as a non-scientist, already recognized how important it is to bring to a wider audience these kind of problems. The problems often hidden in the jungle, not really observed by the official decision maker or ignored. And this guy wrote a book after he wrote a totally different book, which had a lot of, had a very wide audience. Then he utilized his influence and wrote this book, The Last of Their Kind, which was very inspiring. And then an icon of conservation and actually the interface between the public and science is David Attenborough. That is someone who is incredible, and he is he's a kind of half god actually in this field. And I had the privilege to work with him on The Rise of a Mammal, and I'm really admire him. So that's a very important person. And then recently I met the young lady, or interact with a young lady who will play a big role tomorrow on Earth Day. Her name is Ella Luck. It's a little girl, red-haired girl, I guess from England. And I watched some of her statements, and I'm extremely impressed by this young lady, which is a little bit in line with a general development of Friday for Future, that the young generation get faces who speaks out problems which are a long time ignored, and which are their problems because they live much longer than we live. We might have still a reasonable intact planet, but if it goes in this way, then I think we need young people which really care what will happen in the future, and that the lifestyle, the human race now approaching 10 billion people, that this lifestyle is not self-sustaining, that is destructive. So I think there will be a lot of young, activists who are really important for the future of our planet. The old guys have the image, but I think the young ones, most of them are not so well known, like Greta Thunberg, but these young people are really important, and I admire them that they change the way how we grew up, the way of thinking, that not everyone needs a car, but we should utilize fossil fuel, and all these questions which were never asked before because we were impressed by the technical revolution, but the price we pay for that is the price they will then have to deal with in 50 years. That is also something I'm really impressed that the young generation act totally different than we grew up or the generation before.
Excellent points, and a great place to wrap things up. But it's been really eye-opening listening to your story, and as I said, I take my hat off to everything you're doing and in creating these populations, these new self-sustaining populations, and hopefully, we can spread the word a bit and help inspire more generations to do the same thing, because it's so crucial moving forward. But for everybody that's going to be watching on the YouTube channel or listening on the podcast networks, you've been speaking with the amazing Professor Thomas Hildebrandt at the Department of Reproduction Medicine at Leibniz-IZW, Professor of Wildlife Reproduction Medicine at the Freie Universität Berlin, doing really amazing things in cellular technologies, de-endangerment, creating these self-sustaining populations that are going to be extremely beneficial to the future of this planet. Professor Hildebrandt, thank you for what you do. Thank you for the rhinos and the giraffes and the elephants and the panda bears and on and on. But as we say on the show, thank you for moving the human story forward with what you do, because it's so crucial and so important, especially in 2020 with where we are. And thank you for taking the time out of your schedule to come on the show. This was really a wonderful time.
Thank you very much.